So here we could see the extracellular domain of fibroblast growth factor receptor 2 or FGFR2. We could see that there are two chains that make up this domain. These chains form a dimer after the binding of the ligands heparin, which interacts with the FGFR2 at these residues highlighted in red, as well as fibroblast growth factor 1, seen here in orange, that interacts with these yellow highlighted regions on FGFR2. So normally these two chains are further away from each other, but due to the binding of these ligands, these two chains, specifically the D2 domain of the chains, are allowed to come close together, and therefore dimerize. However, this interaction would not be possible without the specific 3D structure that FGFR2 possesses. The right residues need to be in the right location for this specific interaction to occur between the ligands and the FGFR2 receptor, and therefore allow the FGFR2 receptor to become activated. So after dimerization and activation of the FGFR2 receptor, this ternary complex allows for the activation of the intracellular kinase domain that we can see here. So here is the extracellular domain, and this is the intracellular domain at which specific substrates can bind onto at its on its surface. So these specific substrates can lead to specific signaling pathways and therefore lead to distinct cellular responses. So here in part A, we could see the RAS-MAPK pathway that leads to the cellular response of cell proliferation. And in part B, we could see the PI3K ACT pathway that leads to the cellular response of cell survival. So these specific cellular responses would not occur if FGFR2 did not possess the right structure. Therefore, it is vital that FGFR2 has the correct 3D structure that can allow for these ligands to bind and interact and therefore activate FGFR2.